So good day, everyone. Welcome back to the CERC seminar series. I hope everybody had a great summer. My name is Sana and I'm a postdoc in Matt Lawrence's lab, as well as a member of the CERC seminar organizing committee. And today it is my honor to introduce to you our speakers. Our first speaker of today is Dr. Kiran Nakka. Kiran is a postdoctoral fellow in Jeff Dilworth's lab at the Ottawa Hospital Research Institute. And here he studies the regulatory roles of histone modifications as well as modifiers during satellite stem cell mediated muscle regeneration. Before moving to Canada, Kiran obtained his PhD in biotechnology from the National Center for Cell Science in India. Here he studied the role of nuclear matrix proteins and histone modifications in regulation of co-transcriptional mRNA splicing. So today, Kiran will be presenting to us his current work on how temporal demethylation of H3K27 trimethylation by distinct demethylases of the KDM6 family regulate muscle regeneration. So Kiran, thank you so much for being here with us today.
So today, thank you first of all the attendees for coming for this presentation, and thank you CHRC Network for giving this opportunity to present my work that I'm carrying in Dr. Dilworth's laboratory. So today I'm going to talk about my work on temporal demethylation of H3K27 trimethyl marked by distinct demethylases of KDM6 family and how they regulate muscle regeneration. So what we know is that like histone modifications regulate gene expression and contribute to cell fate determination and lineage commitment. And among all the histone modifications, histone H3 undergoes extensive modifications, and these modifications are written by many writer proteins, and then they will be either erased or read by many reader proteins. There are different types of and multiple variants of the same of the proteins that regulate these dynamics. Among the multitude of modifications, the H3K27 trimethyl mark is a hallmark histone modification that regulates cellular homeostasis and embryonic development. And the polycomb and trithorax group proteins antagonistically regulate the K27 trimethyl mark. And here, using satellite stem cells from muscle, we wanted to understand how these two histone demethylases regulate the differential gene regulation. And during muscle regeneration, the satellite stem cells are generally resided under the basal lamina of the myofiber, where they remain in a quiescent state. And upon injury, these cells get activated and undergo self-renewal and expansion, where they differentiate to form a regenerated myofiber. So what we wanted to study is like how the KDM6 family proteins regulate these events of muscle regeneration, because the KDM6 family has the same UTX and JMJ3. They work on the identical histone trimethyl mark, and we saw that UTX and JMJ3 are expressed in the quiescent satellite stem cells, and the major aim of our study is to understand how these two proteins regulate the distinct events of muscle regeneration. So to understand this, we first generated the conditional knockout mice, wherein by breeding them with the PAX7 under PAX7 regulatory gene. And then we studied the regeneration by inducing cardiotoxin damage into the muscle. And what we saw is that the muscle are not efficiently regenerated after the damage. So here, what we understood is that these even though both the proteins work on the same mark, they still cannot compensate for each other, and they regulate distinctive, and they regulate differently to the events of muscle regeneration. So we next tried to understand wherein the muscle regeneration these proteins regulate, and what we saw is that UTX regulates the terminal differentiation step, wherein it regulates the differentiation of the myocytes, whereas the JMJ3 can knock out myoblasts, can still undergo differentiation. And we then looked into the proliferation as well, and we did not see any distinct differences in their proliferation, which indicates that these proteins does not contribute to the events during proliferation. But with the understanding the role of UTX, we earlier showed that UTX distinctly regulates the myogenic regulatory genes during terminal differentiation, where UTX demethylates specific genes such as myogenin, CKM, and all, and regulates the terminal differentiation. So to understand how JMJ3 regulates the events of muscle regeneration, we first performed single-cell RNA sequencing analysis, wherein we deleted the gene by giving tamoxifen and let it recover, and then injured the muscle, TA and gastrocnemius muscles, and then isolated the satellite stem cells after 30 hours and fact sorted them, and then performed the 10x genomic single-cell RNA sequencing to understand the distribution along the pseudo trajectory, the cluster distribution along the pseudo trajectory. And what we found is that in JMJ3 knockout mice, you can see that the cells are mostly accumulated in the early stages of regeneration. So JMJ3 knockout mice, what are found to knockout satellite cells are found to have a problem in exiting quiescent stage. So we verified this activation of the satellite cells by performing in vivo EDU activation assay, wherein we damage the TA and gastroc muscles. And before the cells undergo the first round of division, we injected EDU and then isolated the satellite cells, and then did the EDU activation assay. And here you can clearly see that JMJ3 is required for the efficient activation of the satellite cells, and the cells that are without JMJ3 were not able to exit the quiescent state. And further, we tried to understand whether the defective activation of the satellite cells is whether demethylase dependent or not. For this reason, we generated JMJ3 conditional knock-in mice, which is demethylase dead. And we studied we saw that these mice exhibited defective regeneration after damage. And again, we also studied the activation of these satellite cells isolated from the demethylase dead knock-in mice. And what we saw is a reiteration of the same phenotype, wherein they are not effective in exiting quiescent step. And then when we tried to further understand, delineate the mechanisms, we saw that when we isolated the myofibers from the knockout and knock-in mice, we saw that there is no defect that is coming in these satellite cells. So we thought that this could be coming. What we earlier saw was a defective regeneration, defect in activation, but on the myofiber explants, when we did not see, we argued that it could be because of the inflammatory response that exists after damage. So for that reason, we gave damage, and we injured the mice, and we in one set, in another set we did not injure the mice. So here it goes like after subjecting some mice to injury and some to not injury, we let the mice to recover and develop the inflammatory response for 36 hours, and then isolated the satellite cells and did ex vivo EDU activation assay. And we saw that the mouse that were subjected to injury were basically showing the satellite cells isolated from the other contralateral leg of the mouse that were subjected to injury showed a defective activation, which means that the defect from the JMJ3 knock-in mice is an inflammatory signal dependent. And we further verified this inflammatory dependent activation of the satellite cells by isolating the myofiber explants and from the contralateral fibers of muscles. And then we saw that same phenotype where the cells failed to activate in the absence of, these cells failed to activate in the presence of an inflammatory signal. So we further tried to understand the global gene regulation during this state, and then we performed cut and tag protocol. This is a modified protocol wherein we work with low abundant stem cells after injury, and we modified a previously published protocol from Hanikoff's lab by adapting a spike-in with ampicillin gene. And using this modified technique, we tried to understand the genome-wide K27 trimethyl mark and distribution of H3K27 K27 trimethyl mark and JMJ3 and UTX as well. So what we saw is that, and we also we also did RNA sequencing analysis in the same set of mice by isolating the satellite cells. And what we saw is that we saw both upregulated and downregulated genes in both JMJ3 and UTX. And basically, we concentrated on the downregulated genes as the K27 trimethyl mark contributes towards the activation, towards the expression of the genes. So in the case of JMJ3 knockout, the downregulated genes, we saw a distinct enrichment of K27 trimethyl mark in these specific genes. And we did not work with any of the UTX downregulated genes because UTX mice knockout satellite cells do not show any activation defect. That means these genes does not contribute towards the activation step. And the geo-analysis of the genes revealed that these are the genes that are altered in the JMJ3 knockout satellite cells. And what we saw is that the JMJ3 knockout satellite cells have an upregulated genes for the inflammatory response and the immune response. And there is a increased cytokine storm in the JMJ3 knockout cells. And what we are using CyTOF and analysis, we could see that there are more infiltrating cells in the muscle, inflammatory cells in the muscle after damage, and which is corroborating with our inflammatory response gene upregulation in the knockout mice. And going back to the downregulated genes, we saw around 49 genes that were downregulated and overlapped with enriched K27 trimethyl mark. And one of the genes is the Has2 gene, which is known to modulate the inflammatory signals. So we saw that the Has2 gene is downregulated when we knock out JMJ3, and there is a corresponding enrichment for K27 trimethyl mark and EZH1 as well. So Has2 basically helps in the synthesis of hyaluronic acid here. So the hyaluronic acid basically funnels the signals from the extracellular niche into the cell and helps in the activation of the cell. So what we saw is that in the cells knocked out for JMJ3, there is a decreased deposition of hyaluronic acid at 48 hours post-injury. And even on the myofiber explants, we see that there is a decreased hyaluronic acid around the stem cell niche. And using an inhibitor that blocks the synthesis of hyaluronic acid, we studied how that influences the regeneration and activation. And what we saw is that by employing the 4-methyl-umbelliferone inhibitor, we saw that there is a defective regeneration of the muscle post-injury. And then we studied the activation of these satellite cells in the wild-type mice and the UTX knockout mice, which otherwise activate the satellite cells when you delete the gene UTX. So what we saw is that when you give 4-methyl-umbelliferone to these mice post-injury, we saw that the cells failed to activate, which emphasizes that the synthesis of hyaluronic acid is important for the activation of the satellite cells. And to further understand how hyaluronic acid, whether hyaluronic acid contributes to the activation of the satellite cells in the absence of any inflammatory signals. So for that reason, we isolated the satellite cells from the wild-type mice, and then we treated the wild-type satellite cells with 4-methyl-umbelliferone in the presence of damaged muscle extract that mimic the cytokine signals. And what we saw is that in the absence of any muscle extract, but even though you add 4-methyl-umbelliferone, the cells can activate. but in the presence of an inflammatory cytokine signals, as mentioned, as you can see in the blue bar, that addition of 4-methylaminophorone interferes with the activation, which means the hyaluronic acid is specifically required for the activation of the satellite cells during the inflammatory cytokine signal. So to further establish that hyaluronic acid contributes to the activation of the satellite cells, so we exogenously added hyaluronic acid to the myofiber explants, and then studied the activation of the satellite cells in the presence by EDU incorporation. So in this, as just like satellite cell, we studied the satellite cells, we isolated the myofibers after 36 hours post-injury and then added hyaluronic acid. Here you can see that the knockout myofiber explants show defective activation of the satellite cells, whereas when you supplement these knockout myofibers with the hyaluronic acid, there is a rescue in the activation, which means hyaluronic acid can effectively transmit the extracellular signals into the cell and push it towards activation. So overall, what we saw is that GMGD3 is required for funneling the extracellular signals into the cell via hyaluronic acid, and GMGD3 and UTX temporally regulate the demethylation of satellite cells during muscle regeneration. And GMGD3 specifically modulates satellite cell activation, whereas UTX regulates the terminal differentiation of the satellite cells. And independent of UTX, GMGD3 regulates FAST-mediated hyaluronic acid synthesis and helps in funneling the signals from the niche to the satellite cells. So overall, what we see is that GMGD3 and UTX regulate distinct events of distinct regulatory roles in the same cell type during muscle regeneration, and GMGD3 contributes to regulates the genes involved in immune response and the extracellular matrix genes, probably by antagonizing the EZH1 mark trimethyl mark here, whereas UTX regulates the genes by antagonizing the EZH2 trimethyl mark genes in the myocytes and helps in the formation of multinucleated myotubes, thus contributing towards the terminal differentiation. So overall, I want to thank all my lab members for in this work, and my collaborators as well, who contributed in many ways for this study. Thank you all. I'll be happy to take any questions.
Thank you very much, Kiran. It was great. I think we have time for 1 or 2 questions. If anybody has some, you can put them in the chat, or please raise your hand. I'll get started with one question. I was wondering if you either have a hypothesis or idea about why there is this difference between UTX and GMGD3, or maybe something I don't know about these two.
Yeah, so as I showed in my introductory slides, so these two proteins have a very different protein organization domain. So it could be attributed to the differential protein interactions that they attend over the time. So that could be the reason how each one of them regulate a different set of genes, even though marked with the same trimethyl.
Okay, thank you. And I see that Michael Johnston is raising his hand. So you want to ask your question?
Yes.
Hi, Kiran. Very nice talk. I was curious if the linkage between hyaluronic acid application and like JMJD3 activation is known in like that intracellular leg, because I find it surprising that extracellular HA application can rescue an intracellular JMJD3 deficiency. So how can like an extracellular treatment rescue a genetic defect within the cell?
So basically, when there is especially in the case of muscle damage, when there is a damage, there are multiple pro-inflammatory signals that dominate in the first early hours of damage. So the satellite cells are maintained in a quiescent state during that time and prevent the early exit from the cell cycle before the pro-inflammatory niche is clear. So basically, the hyaluronic acid is probably contributing in the formation of microglobular domains that help in the transmission of these inflammatory signals into the cell and dictate whether the cell to undergo activation or still remain in quiescence or not. So when the cell undergoes activation, so once the cell senses the signal, yes, there is a predominance of a pro-inflammatory signal or anti-inflammatory signal. These proteins probably by undergoing some post-translational modifications and forming a differential interactome, they then regulate the gene expression and contribute towards the cell moving forward, like determine the cell commitment.
Okay, cool. No, thank you.
Yeah, thank you.
So our next speaker is Dr. Jeff Dilworth. Jeff is a senior scientist in the regenerative medicine program at the Ottawa Hospital Research Institute, and he's also a professor in the Department of Cellular and Molecular Medicine at the University of Ottawa. So Jeff obtained his PhD at Queen's University, after which he moved to Strasbourg, France, for his postdoctoral training in the lab of Pierre Charbon. It was here that Jeff started working in the area of epigenetics while he was trying to understand the mechanisms regulating steroid-induced gene expression. Then after his time in Europe, Jeff moved back to North America and he continued his training with Steve Tapscott at the Fred Hutch in Seattle. And here he examined the role of epigenetics in muscle regeneration, a topic he's still very interested in today. So today, Jeff will be talking to us about how transcriptional control of muscle stem cell expansion ensures muscle repair and niche repopulation. So thank you so much, Jeff, for being here with us today. And please take it away when you're ready.
Great. Thanks, Sunny, for the introduction. It's a pleasure to be here today and to be able to tell you about these exciting stories we have in the laboratory. Great job, Kiran, with your story there. So I'm going to tell you a different story that we just published in the laboratory, which is about the transcriptional control and the negative elongation factor, how the transcriptional pausing is affecting the ability of the stem cells to be able to repopulate the niche as well as repair the muscle fibers. So first of all, I'd like to take a second to be able to tell everyone who doesn't follow muscle all the time that muscle is in fact a very regenerative tissue. And if you have a catastrophic injury to the muscle, either a freeze injury, it could be a snake bite, you're going to have extensive damage to the fiber. That you can see the healthy fibers over here on the left. Once you have damage, you're going to have necrosis of that tissue. You're going to have then inflammation. All of the different cell types from the blood are going to make their way into the tissue. The macrophage are going to remove the damaged myofibers. Eventually, you're going to get activation of your satellite cells, your muscle stem cells, and they're going to carry out regeneration, and you're going to have remaking of these myofibers 14 days later. And we can tell the difference between a regenerated myofiber and an undamaged myofiber by the presence of the nucleus here in the center of the myofiber versus the nucleus being on the periphery over here in the healthy myofibers. Okay. So the cells that are responsible for carrying out this regeneration are the satellite cells over here on the left. And normally they're going to be present in a quiescent state, but they're going to become activated in response to injury. They're going to proliferate to give you enough cells to be able to remake the myofibers. Once you remake these myofibers, you also have to be able to repopulate the niche so you can be able to repair the tissue if there are future damages. Now, I'm going to tell you why I really enjoy studying satellite cell biology and the epigenetics of this. Is the idea that satellite cells are made during embryogenesis. So you can see tearing away here from the dermomyotome, you've got these progenitor precursor cells here that are migrating. These are actually satellite cells that are going to move into the limb bud and they're going to make your muscle. Now, a study that came out a few years ago from a colleague in Paris, they went into, they did this both in mice and in humans, and the picture I'm showing here is from mice, but they took old-aged mice or people who had died after a natural death between 90 and 100 years of age. They isolated the satellite cells from these either mice or humans several days after they'd already been dead, so hypoxic conditions. They can take those satellite cells, inject them into the mice, and they're able to regenerate. And here you can see that those cells that were taken from the dead mouse are transplanted in and are able to give rise to new myofibers. So what that tells us is that these cells are there throughout your lifetime, and there's no reason why we have to be losing our muscle mass as we get older. Now, this here is just showing that, of course, we are losing some of the stem cell numbers as we get older, but there are still lots of stem cells left. But as we get to be 65 years old, what we see is that there's a loss of stem cell function. So why is it that those stem cells are not able to respond as well? Now, thinking about this image here that I'd already shown you before, each one of these cells has the same DNA in it. And as we move through the different cell fates, we're going to change the set of genes which are being expressed. And of course, this is regulated at the epigenetic level. When we're young, this is a very efficient process. And when we get older, this becomes inefficient. And why is it becoming inefficient? Because we're changing the way that the chromatin is compacted, and that's going to affect the genes which are going to be expressed. Now, of course, epigenetics being something which is reversible, I think that there is the potential that we can reverse these changes that we're seeing through changes in lifestyle and dietary changes in order to make these stem cells young again. So that's really what gets me going about the muscle regeneration and epigenetics. Okay, so what I'm going to talk to you about today is a story that we looked at that has to do with proximal promoter pausing. And of course, in regulation of transcription, a lot of us look at the recruitment of chromatin remodeling factors, which are going to allow the general transcriptional machinery to bind to the promoter, and that's going to activate transcription. But what happens at a lot of developmentally regulated genes is that the polymerase gets recruited to the promoter and moves into the gene, but actually pauses about 20 to 60 base pairs in. And we've got a Nelf complex here, which is associated with a negative elongation factor. And the role of that negative elongation factor is to stabilize the RNAs that are associated with the polymerase. So the polymerase is going to be idling, moving back and forth along the DNA, and that's going to cause kinks in the RNA, which would then be accessible to endonucleases and result in abortive transcription. So in order to stabilize those transcripts, we've got the Nelf protein. And when you want high levels of expression off of the gene, you get pause release. Sorry, hopefully that's not going to happen again. So we get the pause release happening here, and Nelf goes away, and we get high levels of gene expression happening. Okay. So Nelf is a 4-protein complex here that binds onto the polymerase and competes with the elongation complex, the super elongation complex, for positioning on there. And if you knock out any one of these 4 subunits, you lose the activity of all of the complex. So we wanted to know if we had a role for Nelf and promoter proximal pausing in muscle regeneration. And to do this, we went and knocked out Nelf specifically in the muscle satellite cells. For this, we went and obtained from Kiran Nakka Nelf floxed allele, and we crossed that into mice that had PAX7 expressed CRE-ER. So only in the satellite cells, which express PAX7, they're going to express Cre-ER. When we add tamoxifen to those cells, the Cre-ER is going to enter into the nucleus. It's going to remove Nelf, and we get the deletion specifically in the muscle satellite cells. So the first question we wanted to ask is, what would happen if we didn't have Nelf in the cell when we carried out a muscle injury? And we got these surprising results here. If you look at the wild type and the knockout underneath it in the cardiotoxin treated leg, the first thing, after 28 days, it looks like they regenerated very well. There's not an overt problem with that muscle, except for the fact that there's a hypertrophy, which means that these myofibers are a lot thinner than in the wild type condition, and that's quantitated over here. And we didn't give much thought of it until we look at this picture here. The normal regenerated fibers look quite like the undamaged, whereas we've got these very tiny muscles being regenerated in the absence of Nelf. And to try and understand the hypertrophy, the idea is that the myofibers are smaller because they don't make enough protein. So we went and we isolated some myofibers, and we looked at the spacing of the nuclei along this multinucleated myofiber, and you can see that the spacing has doubled between these nuclei. And what that means to us is that there's half as many cells fusing into the fibers in order to make up those, able to make up those regenerated myofibers. So what happens to the satellite cell compartment after these muscle injuries? We went, and hopefully you can see on your screen these red cells. These are the satellite cells being marked by TD tomato. And what we look at when we start counting them across the entire muscle is that the repopulation of the stem cell niche at 7 and 28 days is greatly impaired in these muscles that don't have Nelf. So not having repopulation of the niche, what's going to happen if we carry out a second muscle injury? Here we went and we injured the muscle once at day 0, a second time at day 28, and we sacrificed the mice at 56 days. What you can see is you get excellent regeneration in the wild type mice, but here there's almost no regeneration taking place in the Nelf knockout. And if we look at the whole muscles, you can see the huge difference between the wild type and the knockout here. Now, the nice thing about the muscle regeneration system is we can look at it in vivo, but we can also take this out into cell culture and look at either the myofibers as an ex vivo system, or you can put them in culture and look in vitro at the ability of these cells to undergo the different stages of regeneration. Of course, as Kiran said before, you've got the activation of the satellite cells, the expansion of the cell population, and differentiation. So the first thing and the easiest thing to look at is differentiation. And we didn't expect that there was going to be a differentiation defect, because in the absence of Nelf, we were able to generate these nice looking myofibers, even if they were a little bit smaller. And when we put in culture these cells and carry out differentiation, we get excellent formation of multinucleated myofibers. So there's no defect in these Nelf minus cells in their ability to differentiate. Now, Kiran just showed you an activation defect for the JMJD3 knockout mice. So we went and did a similar experiment where we did first a cardiotoxin entry to the TA muscle, and then we would inject through the interperitoneal area EDU at 28 hours, just before the cells would enter into cell cycle. And we give them 12 hours to enter into cell cycle, and we look at the activation. And we can see that there's no significant difference in the ability of these cells to enter into a cell cycle. However, if we wait 60 hours, so after 40 hours, when they first enter into cell cycle, they start to replicate every 8 to 10 hours. So they're cycling very quickly. So what we did in this experiment here was to look 60 hours after injury. We did an EDU pulse into the IP area and recovered these cells at 72 hours. And here you're able to see a significant decrease in the ability of these cells to proliferate. Now we can take these cells out and look at them growing in culture. And again, If we do an EDU assay to look at the passage through S phase of the cell cycle, this is greatly reduced in the Nelf knockout. And if we just count the number of cells that are accumulating on a plate over 3 days, you can see that there's very, very little proliferation going on there. Sorry. So the next thing that we wanted to look at was using single-cell RNA-seq, whether there was going to be a disruption in the population of cells. And what we see here is that there's 2,000 cells being plotted out over the trajectory, and we were able to break that down into 15 different clusters. Using pseudo-time reconstruction, we can see that they start over here. These are PAX7 positive stem cells. They go into a proliferative stage up here, and these are myogenin positive cells that are ready to undergo differentiation. We're only working with mononuclear cells, so we don't actually see the differentiation. And from this experiment here, what we found was that almost all of the wild type cells, because this experiment was done 3 days after injury, almost all of the wild type cells are in the proliferative stage up here, whereas in the Nelf knockout, a lot of the cells that were in the proliferative stage have made their way out into the pre-differentiation stage here. And if we look at the genes that are differentially expressed across these clusters, what we see is that we've got genes involved in cell cycle and also p53 signaling being effective there. So next, what we did was we carried out bulk RNA sequencing, and we did this at 48 hours after injury, because now we know that the problem in the regenerative process is at the stage where the cells have just entered into cell cycle and are starting to proliferate. And so at 48 hours, they should be in the state, just entering into the state where they're going to be expanding at a very fast rate. And what we can see is that we've got upregulation of things like MAP kinase signaling, which is a signal which drives the cells towards terminal differentiation. And we've got decreased expression of genes involved in cell cycle. And the same sorts of genes are being regulated in the ex vivo expanded cells. So we wanted to know whether proximal motor pausing was contributing to this. And so we collaborated with Kiran Nakka at Harvard on this, where she did ProSeq analysis, where you look at the formation of nascent RNA formation by using these biotinylated nucleotides here. And we look for accumulation of transcripts close to this TSS area. So when we looked at all of the different genes that are regulated by Nelf, we did not see significant change in the upregulated genes for their movement into the gene body. We do see a little bit of an increase in the absence of Nelf of the RNA polymerase moving into the gene, but that's not statistically significant when we calculate it over here. But where we do see a statistically significant effect is at the downregulated genes. So we're losing transcript stability at these pause genes when we knock out Nelf. Now we went and we combined our RNA-seq data, both in vivo and in vitro, with the ProSeq data, as well as cut and tag analysis for Nelf and ChIP-seq analysis for H3K4 trimethyl, as well as phosphorylated polymerase. What we can see is this PEDF, or serpine F1, which is a growth factor, is strongly downregulated, both in vitro and in vivo. And we see a loss of the stalled polymerase here at the transcription start site, and Nelf goes away from that site as well. And we lose the accumulation of the polymerase without really affecting H3K4 trimethylation. Now, PEDF is a signaling molecule that affects p53 signaling. So when you have PEDF, you get signaling down into the nucleus, which is going to lead to the expression of cyclin G1, cyclin G1 is going to affect the stability of P53 by sequestering the phosphatase here and preventing the MDM2 from being degraded. So in this case here, when we have the PEDF, you're going to have you're going to have degradation of the P53, and these cells are going to continue in cell cycle. However, when we don't have the PED53, we're not going to be making as much cyclin G1. That's going to allow P53 to accumulate in these cells, activate P21, and the cells are going to exit the cell cycle. At least that's what we were thinking. So to test this, we went and we treated cells with an inhibitor of P53, the P53N here. And this experiment here was done in a way that we injected cardiotoxin to cause the muscle injury. And then starting at 48 hours after the cells have entered into the cell cycle, we did intraperitoneal injection of this P53 inhibitor 3 times. So it's not going to inhibit all of the P53 activity, but it's going to reduce P53 activity in the cell. And what we were able to see with that is that the P53 inhibitor was able to increase the size of the myofibers in the Nelf knockout. And we also saw that there was an increase in the number of PAX7 positive satellite cells that are associated with the regenerated fibers. We also looked ex vivo and in culture, and what we could see was that if we treated satellite cells, either the wild type or the Nelf knockout, with either PEDF, or the P53N, what we saw was normally you get the knockout having poor incorporation of EDU. They're not proliferating as much, but that is rescued either with the PEDF or the inhibition of P53. So what that tells us is that the PEDF P53 signal is contributing to the proliferation defect in this Nelf knockout here. Now, we know from these experiments that I just showed you that Nelf is necessary for preventing the cells from moving from this proliferating progenitor stage into the differentiating myotubes, and is important for the expansion of this progenitor population here. But we're also seeing a defect in the ability of the stem cell niche to be repopulated. So we wondered whether Nelf might be involved in this process as well, the return to quiescence. Now, based on current models for how the stem cell niche repopulates, what would happen is that the quiescent stem cell would become activated. It's going to undergo its first cell division. And what would happen in an asymmetric cell division is one cell would commit to the muscle lineage and go into the proliferating progenitor population, and the other one would remain a stem cell and repopulate the niche. Now, our data is not consistent with this up until now because we didn't see any defect in the activation step. It was only after 40 hours that we started seeing this defect, but we wanted to test that possibility. The other possibility is that this repopulation of the niche and the quiescence population over here on the left would happen due to a stochastic mechanism where, let's say you made 10,000 of these proliferating progenitors, you would use 9,950 of them to differentiate to form these myofibers, and 50 would go back to repopulate the niche. And if we had a defect in our proliferation, maybe you only make 400 cells, and there 398 are going to go to make the fibers, and there's very little of them to return to the niche. To test these two different possibilities, we did transplantation studies. So we irradiated NSG mice, and we isolated satellite cells that were TD tomato positive, either from the knockout of Nelf B or from wild type mice. We put 12,000 cells, satellite cells from each, into an undamaged muscle and allowed 21 days for those engraftments to occur. And then we looked at the engraftment into the muscle. And the first thing you're going to see is that there's a lot more muscle that is marked by TD tomato here in the wild type compared to the Nelf. And this is expected because of the fact that these cells that we inject are going to proliferate. So even though we put 12,000 into both of the mice, that number is going to greatly expand in the wild type compared to the knockout. So we weren't too concerned about that, and that's the quantitation here. But when you blow this up and you just look at the repopulation of the stem cell niche on those muscles that have turned red, meaning that they've got satellite cells in their vicinity, what we see is that the same percentage of TD tomato positive myofibers are also positive for PAX7 positive TD tomato cells. So the repopulation of the niche is happening with these satellite cells that had already entered into cell cycle. And what that tells us is that the satellite cells can repopulate the niche, first in the absence of Nelf, and secondly, that the repopulation of the niche is happening through this second mechanism of the progenitors returning to a quiescent state rather than an asymmetric cell division taking place during the first cell division. Okay, so just to finish off, I'm going to give you this model here that Dan from my lab put together. So normally what's going to happen in a wild type cell is you're going to have stalled polymerase due to Nelf, and when you get the muscle injury, the satellite cell is going to become activated. It's going to start proliferating. It's going to continue proliferating until you have enough cells to repair the myofiber and also to repopulate the niche. Okay. Now, in the Nelf knockout, you are not going to have a large expansion of these cells. The poor expansion is going to have fewer cells fuse into the fiber. You're going to get a much more narrow or a smaller diameter myofiber, and then you're not going to get repopulation of the niche. So what I've shown you here today is that RNA polymerase pausing is a key mechanism used in satellite cells to prevent premature activation of the p53 signaling, and that when we don't have this stalling occurring, you're not expanding your stem cell population sufficiently, first of all, to repair the myofibers. But not only that, you don't have any stem cells left to repopulate the stem cell niche. And then what the more important in terms of stem cell biology take-home message from this is that the satellite cells are not repopulating the niche primarily through asymmetric cell divisions. And I'm not saying that the asymmetric cell divisions don't happen. They're likely happening at a very low rate, but the predominant mechanism used to repopulate the niche is this mechanism of expansion of the muscle progenitors and their return to quiescence. So just as a last slide, I want to present this, which sort of brings together the data from Kiran's talk and my talk. And of course, we're talking in these different presentations about ubiquitous transcription factors or epigenetic factors. We talked about JMJD3, we talked about Nelf, we talked about UTX. Every one of them is present in the cells at each one of the different identities. And they're affecting gene expression if you knock them out. UTX and JMJD3 both affected about 325 genes when they were knocked out in the satellite cells, but really JMJD3 was what was contributing to the regeneration. So we've got each one of these different steps being regulated by different epigenetic or transcription factors. And what that tells us is that there's the potential for going and carrying out epigenetic therapies that are going to allow for being able to better activate the satellite cells, or for expanding these populations more, or causing them to differentiate a little bit better. And just a thought about epigenetic therapies, because people are always saying, well, you have to be very worried about off-target effects because the UTX inhibitor is going to affect all the different cell types in the body. That's not really true, because we show that UTX is not necessary for any of these early stages here, only for the terminal differentiation. And in fact, we have the knock-in mouse that doesn't have any H3K27 activity, and we're able to breed it as a as a homozygous mouse and have multiple generations. So that H3K27 activity is not absolutely necessary, but we do know that those mice, if we cause an injury and they're not able to remove H3K27, they're going to have poor regeneration, but eventually they will overcome that and regenerate the muscle. So with that, I'm going to end and thank people who did the work. The whole project that I just talked about there was carried out by Dan Robinson in my laboratory. Kiran helped him out with the cut and tag experiments here. And of course, we had great collaborators, Karen Adelman's group at Harvard, who supplied us with the original Flox allele, as well as carried out the ProSeq analysis. And we have collaborators in Lyon who helped us out with defining the phenotype initially. So thank you very much, and I'm happy to take questions.
Thank you very much, Jeff. That was a very clear presentation. I learned a lot. If there's any questions, so please put them in the chat or raise your hand if you want to ask them. That was just a stop screen share. Yeah, I have to say that I'm still struck by what you mentioned in the beginning, how these stem cells were still able to function after like being in a dead subject for 10 days. That still strikes me. It's amazing.
But these cells have to protect themselves against all of the different environmental stresses throughout their lifetime. So the niche that they live in is protecting them, that they're not exposed to so much of the damage. And I think that the fact that they're able to live without oxygen, that's a little bit interesting. That I wouldn't have suspected that, but I'm just fascinated by that paper that came out 105 years old and isolating satellite cells and able to put them into a mouse and they make muscle. That's
very impressive. There's no questions yet in the chat. So I'm very new to this whole field of muscle cells. And I was just wondering if this absence of Nelf, does it actually also have an effect on the expression of PAX7? Because that seems to be a very important gene here. Did you look at that?
Yes, so we spent a lot of time trying to figure out where the defect was going to be. So we did RNA sequencing at many different time points. And of course, things like PAX7 and MyoD and myogenin are the genes that we're going to look for immediately. And PAX7 expression is not where we didn't see any defect there. We actually went into these experiments thinking that there was going to be an activation defect. That was our initial hypothesis. And the fact being that these cells are in quiescence and they have to respond to the muscle injury very quickly. So we thought that maybe we would have the polymerase sitting on all of the genes that are necessary for the activation, and that once the signal started coming in from the myofibers to say that there was damage, that they would just release from that pause position and enter into high level of transcription, but that's not at all what we saw.
Mm-hmm.
Yeah, it seems to be highly specific to regulating the proliferation. And P53 is one pathway which contributes to it, but I'm sure we've got a lot of other signaling pathways as well that are coming into it. Nelf seems to be playing the role of controlling the expansion of the cells, P53 being one of the pathways that would come through.
Yeah. I see that Sheila raised her hand. You want to ask a question?
Hi, thanks. Great talk. I was curious to see that it's really only the proliferation part that's affected. Do you think it's more like a function of timing, like that the lack of Pol2 pausing just can't keep up with the fast cell cycle, rather than any specific effect by the Pol2 pausing itself?
So we are seeing decreased expression of these genes, which suggests that the Nelf, I should point out that we're not actually looking at the pausing. We're looking at the stability there because Nelf is stabilizing the transcripts from the paused polymerase. And what we're seeing is that in the absence of Nelf, you can still have pausing occurring there, but the transcripts are resulting in abortive transcription there. If we were going by the mechanism of pausing not happening, we would see the movement of the polymerase into the genes. Okay, and our ProSeq didn't suggest that. Our ProSeq is suggesting that we're not seeing a massive movement in, and that's consistent with Ali Shalatifard's work, which suggests that Nelf is not blocking the movement in. It's really there to protect the transcript. So there are other mechanisms, including the +1 nucleosome, which are preventing the polymerase from moving in. So with that being said, I think that our data is suggesting that it's really a problem of transcript stability as opposed to not being able to keep up with the cells that are proliferating and having the pausing not take place.
So thank you, everyone, and have a great day.